Hi, this is Dr. Allen. We're going to talk now about various ways that things could be transported across the cell membrane. This transport can either be through something called active transport or passive transport. It could be by diffusion, whether it's simple or facilitated. We're going to talk about osmosis and tonicity as well, and we're going to talk a little bit about this thing called a sodium-potassium pump. Let's go ahead and get started. So just a comparison between passive transport and active transport we see listed here. For passive transport, the key here is that there's no added energy required. Okay, compare that to active transport. It requires energy, usually in the form of ATP. Although there are other mechanisms of energy in cells, but we're not going to really talk about that in this course. Right now, just focusing on ATP. And then in passive transport, substances move down their concentration gradient. It goes, in other words, from an area of high concentration to low concentration. On the other hand, active transport, substances will move from a low concentration to a high concentration gradient. In other words, it goes against their concentration gradient. That's important because that's why you would need this energy in order to help that process go through. I highly recommend you watch this short video clip in your text. It's about 4.5 minutes long. It's in section 3.3, and it has some really good illustrations and explains very well membrane transport, probably a little bit better than I could. So I would defer you to take a look at it through the book. It's nice imagery as well, nice animations. So be sure you take a look at that as well. In the meantime, though, let's look at this here, cell membrane diffusion. And so we can kind of follow the steps here on the diagrams. So step one, we have a insertion of a little pellet. This is a little purple dye. It's inserted into that beaker of water. This is considered an area of high concentration, and the area around it is considered low concentration. So what happens is that through kinetic activity, through thermal motion, the cells of the substance in the dye spreads out to the point where it's evenly distributed, so that there's no longer an area of high or low concentration. It's all relatively equal throughout the beaker, right? So there is no longer a concentration gradient once it finishes. So when we're talking about diffusion across the cell membrane, where this here would be the cell membrane, right? This is that phospholipid bilayer. We have the hydrophilic heads and the hydrophobic tails. You can have something called simple diffusion. Basically, a simple diffusion means that in this case, these little lipid-soluble solutes, these little yellow dots here, kind of scattered about, will diffuse across the cell membrane into the cytoplasm until the point that there's no longer a concentration gradient, right? So until it gets to be that the little particles on the inside are in the same concentration of the particles on the outside, just kind of like that beaker of water and the dye that kind of goes through there, right? The reason why these lipid-soluble molecules can pass through the membrane so easily is because of this phospholipid bilayer, this hydrophobic, I was going to confuse hydrophobic tails, but lipids, other fats, can easily pass through. So that's simple diffusion. Then you have facilitated diffusion. Facilitated just basically means you're helping it. Okay. So this facilitated diffusion, again, no ATP is necessary for this, but this particular example here, we have this membrane protein. It has a particular shape of it. You have these, say, these sugars or amino acids kind of sitting out here. These guys are lipid insoluble, means that the particles themselves aren't able to pass through that membrane. Okay, so it has to get in somehow. Well, one way that I can do it is it can use this particular channel protein. I'm sorry, not a channel. It's a carrier, not a channel. This guy over here is a channel. But anyway, so it can use this protein carrier. It'll go into the protein carrier. Once it goes into there, that will trigger the shape of the protein to change and push the molecule through. Once it does that, then it'll go back to the original shape over here, take another one, and it'll help get it through. Carrier-mediated facilitated diffusion. This other guy over here, this letter C on this right side, is a channel-mediated diffusion, right? So this protein membrane, not protein, this protein in the cell membrane is basically, it's a channel that will allow a particular substance, in this case these little lipid insoluble particles or solutes, remember, because they're insoluble means they can't get through that plasma membrane. Instead, they'll just pass through the channel here like so. It'll allow these little particles to come through, but it won't allow, say, these particles to come through just based on the shape that those particles are. Next, we have the concept of osmosis. We're going to dive a little bit deeper into osmosis at the lab portion. But essentially, one of the big differences of osmosis and diffusion is that when we're looking at diffusion, we were looking at things that were particles, solutes, the little dots that are floating around in the water. When we're talking about osmosis, we're really just talking about the movement of water, not the solutes. Okay, so for osmosis to occur, you have to have what's called a semi-permeable membrane. Now, in this case here, that's this membrane right here in this example. They call it selectively permeable. It's just kind of the same thing, semi-permeable or selectively permeable membrane. What that means is that some things will be able to pass through that membrane, but other things won't. It's just kind of like that cell, right, we were talking about. In the previous examples of the transport molecules, some things like lipids can pass through the membrane, other things can't pass through the membrane. But in this case, for osmosis to occur, you have to have a membrane where water can pass through it, but other molecules, like in this case, a sugar molecule, can't pass through it, right? That's what it's saying right here. Water can pass through the membrane, but the solute cannot pass through the membrane. So you can kind of see in this example here, on this side of the tube, you got a bunch of solute relative to this side of the tube, where there's relatively fewer solute particles. It means there's a higher concentration of solute on the right side compared to the left side. So what is the tendency of water to do? Well, the natural tendency is for there to be a same concentration on the boat on either side. So water will be going in this direction across that membrane in order for it to be balanced. Okay, so it'll say that both sides have the same, what they say, osmolarity. That's the dots that's in the water, but they're going to have a different volume because the water is going to move over here compared to this side over here where it was like the same level. But the tendency of the water is to go onto this side to make that concentration fairly equal and balanced. Okay, so now we're going to talk about tonicity and kind of give some examples of where the water will flow via osmosis. Okay, so in this example here, I have a beaker of water, and there's, but just say we'll call that a cell. It has a semi-permeable or selectively permeable membrane in that water can go through it, but these black dots, the solute, cannot go through it. And so the term of hypotonic is talking about the tonicity of a solution, and that's kind of where we talk about how much of of the particle is dissolved in the solution compared to the cell, right? So in this case, we're talking about a hypotonic solution. Hypo means low. Okay, so the solution itself is hypotonic, meaning that there's more particles inside of the cell compared to outside of the cell, right? Hypotonic, the solution is hypotonic. Relatively dilute is another way that you can say it. So in this case, through osmosis, which way do you think the water will flow? If you said into the cell, you are correct, right? Because remember that previous example, the water wants to flow into um, the cell or whatever structure in order to balance out the tonicity of the two solutions. So it ends up being what we call isotonic. And I kind of drew like, 4 dots on the inside, 4 dots on the outside to show the concentration is the same inside and outside. So isotonic. Now, with this isotonic solution, where will the water flow? Well, at this point, it's achieved a balance. So that means that the water is going to be flowing in and flowing out at about the same rate so that it's going to be very stable and consistent because the tonicity, the amount of solute dissolved in the beaker and inside the cell are the same. Now we're going to have a solution that's called hypertonic, right? So you see in each instance the cell has 4 dots in it. But in this case, there are many, many, many dots on the outside of the cell that's in the solution. So the hyper means excessive, right? So the solution is hypertonic compared to the cell. That means there's more of these little dots in the solution compared to the cell. Now, which way will the water flow in this case? Well, if you guessed out, you are correct, because the tendency is for the water to want to leave the cell in order to balance out the tonicity of the solution so that it looks more like this guy here in the middle. It wants to be isotonic. So that the water is going to want to be pushed out of the cell through osmosis. Now this is from a book, but it's a very, very classic example of how cells react based on the tonicity of a solution. So here we have an isotonic solution, right? This is a red blood cell. This is a normal red blood cell. And so you can kind of see here how the arrows are going in both directions. The solution on the outside of the cell, the tonicity is the same as the solution on the inside of the cell, or the contents of the cell. So the cell is nice and balanced and it's happy and everything is great. Now, in this case over here, letter B, we have the cell is in a hypertonic solution. That means there's a lot more of those dots here on the outside. So the cell will lose water. The water will leave the cell to try to balance out that tonicity. It wants to make it isotonic. So what ends up happening as the water leaves the cell, well, the cell will kind of shrink and kind of shrivel up because all the water is leaving it. In comparison, we have a hypotonic solution. Okay, and so the water is going to want to go into the cell. Remember, because in a hypotonic solution, there's more particles inside of the cell than outside. So the water is going to want to try to go into the cell to balance it out. But as it does that, the cell will expand and expand kind of like a balloon, and it could eventually pop. Or in this case, as it says here, it can lyse. Lysis, remember, lysis means destruction. All right, so those are passive transport mechanisms. First, we talked about diffusion, which is like uh, particles going across the membrane. Then we talked about osmosis, which is water going across the membrane. But in terms of active transport, we're going to talk about things where you need to have energy for substances to cross the membrane, and that energy is typically in the form of ATP. The other thing about this, as we said before, this can go against the concentration gradient, so it can go from low concentration to high concentration. So when we're looking at active transport, one example of that is called primary active transport. And primary active transport, an example of that is what's called the sodium-potassium pump. That's going to be very, very important when we're talking later on in this course about things like action potentials and excitability of cell membranes, in particular when we're talking about nerve cells and nerve transmissions and that sort of thing. But for now, let's just kind of touch on the subject by saying that for this sodium-potassium pump to work, this membrane pump, it's a protein, you need to have ATP, and the ATP phosphorylates that transport protein in order for it to work. Okay, so when we're talking about active transport with the sodium-potassium pump, I'd like you to take a look at Focus Figures 3.2 in your text. It has the actual full diagram in one piece. I had to break it into smaller segments so that you're able to see, at least on the screen here, what they look like. So the sodium-potassium pump, the end result of this by use of one ATP molecule, and we'll see how that works, but one ATP molecule will result in 3 sodium ions to be pumped out of the cell, and then 2 potassium ions to be pumped into the cell. In the end, what that ends up meaning is that there's lots more sodium on the outside of the cell compared to potassium on the inside of the cell. But that's just kind of the normal balance that creates a normal electrochemical gradient that will make more sense later on in this course when we're talking about action potentials and membrane potentials. But for now, let's just look at how this pump works. Okay, so here's this plasma membrane. Here's a protein. It is a transport pump. Here's the step one. We have within the cell, these yellow dots, the sodium will bind this protein. Okay, it'll bind into there. Now, when that happens, sodium binding will promote the hydrolysis of ATP. And when that happens, it releases energy, and that phosphorylates the pump. Right? So ATP is attached to the pump here. When those sodium binds onto there, it'll hydrolyze this ATP, which will result in ADP, leaves that phosphate group onto the pump itself. It phosphorylates the pump. Next, that phosphorylation causes the pump to change shape, opening it up like this. You see now the sodium goes out to the outside. See now these little yellow dots, the sodium will go to the outside, and that's a result of that phosphorylation event right there. Now when that happens, now the pump is open. Potassium, these little green dots, will now bind to the inside of the pump here. Right, 2 potassiums will bind into there. Okay, so now this step we're over here. Potassium binding here will release the phosphate from that right there. When that happened, the dephosphorylated pump, the phosphorus comes out, resumes back to this configuration here, right? Because before it was open up to the outside, the potassium binds, releases that phosphate, and then it comes back to this position here. And then the final step here, because there's no longer a phosphate attached to it, another ATP molecule comes here, attaches to the pump. When that happens, the potassium comes inside. Now it goes back to that first step that we were on before, where the sodium, the yellow dots, can now go into there. and then the cycle returns again. So that's that sodium-potassium pump. Through the use of one ATP molecule, we'll have 3 sodiums leave the cell and 2 potassiums coming into the cell. And then just remember that this generates what's called an electrochemical gradient across that membrane of the cell. Okay, that's it for this lesson. We shall see you for the next one. Take care.